blood group terminology and common blood groups. So for an introduction, accurate communications regarding red blood cell antigens, antibodies, genotypes, and phenotypes does require the correct use of naming conventions. So currently there are 346 red blood cell antigens in 36 systems, and they are formally recognized internationally. There are chemical treatments of red blood cells that provide a tool for antibody identification as well, and we'll talk about all of that. So a blood group system consists of one or more antigens produced by alleles at a single gene locus or closely linked loci wherever or where crossing over is rare or absent. Genetic variations can include co-dominant alleles, so that's where both alleles express simultaneously. Silent or amorphic alleles, so this is where alleles that produce no antigen. Null phenotype, so that's the absence of all antigens in a system. High prevalence antigens, so these are antigens that are present in most individuals. And then we have regulator or modifying genes, so these are genes that influence expression of blood group antigens. Genes, antigens, and phenotypes follow specific naming rules to avoid confusion. Gene names are usually italicized and may use uppercase or lowercase letters. Antigen names are typically non-italicized and often will use uppercase letters. And phenotypes are described using antigen names or even abbreviations. So we do use subscripts, superscripts, and formatting like italics, and this helps to distinguish between genes, alleles, and antigens. So A plus, with that plus sign, or a minus, the minus sign, next to an antigen indicates whether the antigen is present or absent on the red blood cell. Antibodies are named by adding the prefix anti to the antigen name with a hyphen before the antigen symbol. So for example, antigen D is anti-D and antigen K is anti-K. The International Society of Blood Transfusion, or ISBT, formed a working party on terminology for red cell surface antigens back in 1980. Its purpose was to standardize and enable communication on computer systems. So each antigen is assigned a 6-digit number. The first 3 digits identify the blood group system, collection, or series, where the last 3 digits identify the specific antigen. To date, there are 30 blood group systems that have been recognized. Collections consist of antigens that have biochemical, serologic, or genetic relationships but do not qualify as a full blood group system. Antigens and collections are assigned numbers in the 200 series, so those numbers that start with 200. Some other red blood cell antigens, so red blood cell antigens not part of any system or collection are then cataloged into 700 series, which are low prevalence antigens, our 901 series, which are actually the high prevalence antigens. The Lewis blood group system. So Lewis antigens are not intrinsic to red blood cells, but they are present on type 1 glycosphingolipids that are passively absorbed into or onto the red blood cell membrane from plasma. The main antigens are Lewis A and Lewis B. And then we have genetics. So the Lewis locus, there are 2 alleles, capital L, lowercase e, which is the functional, and the lowercase l, lowercase e, which is the amorphic, are non-functional. They also have the secretor locus, so there are 2 alleles with the capital S, lowercase e, which is, again, the functional, and lowercase s, lowercase e, which is the amorphic or non-functional gene. For occurrence, these are often naturally occurring in Lewis A negative or B negative individuals when they have no known red blood cell stimulus required. They are generally an IgM class, and they do not cross the placenta. For fetal red blood cells, antibodies are not well developed on fetal red blood cells, so it's not much of a concern for them. For common types, we have anti-Lewis A, which is the most commonly encountered Lewis antibody, and anti-Lewis B, which is the less common and generally weaker than the anti-Lewis A. The development of antigens for Lewis antigens, so saliva, Lewis A and Lewis B glycoproteins can be present in the saliva of newborns depending on inherited genetics or genes. For plasma, the Lewis glycolipids are not detectable in plasma until about 10 days after birth. The P blood group system, so the ISBT assignment, the P1 is assigned to the P blood group system, or the 003, which is located, as you see on the slide there. And then the P, just the regular P, is assigned to the globocyte blood group system, which is the 028 number. Capital P, little k, and capitals LKE are assigned to the globocyte collection 209. Some common phenotypes for the P group is P1 and P2. So anti-P1 antibody in nature, this is common. It's naturally occurring IgM antibody found in sera of P1 negative individuals. For reactivity pattern, that's usually weak and cold reactive, and it's not often seen in routine testing. For cross-matching, acceptable approach, so if the cross-match is compatible at 37 degrees Celsius and antiglobulin phase is negative, it is safe to go ahead and transfuse this. You may require incubation at room temperature or even an enzyme pretreatment to enhance reaction if it is suspected in the blood. For clinical significance, it can cause hemolytic transfusion reactions and hemolytic disease of the newborn, but it is really rare. And it can be IgG, but usually it does not cause hemolytic disease of the newborn. Once again, this is usually in IgM. Cold agglutinins and big I, little I antigens. So cold agglutinins can be present in serum of normal individuals and in patients with acquired hemolytic anemia. The capital I and lowercase I antigens are important in this context. So we can use enzyme treatment like Ficin and papain, which enhances the reactivity of uppercase and lowercase I antigens. Anti-A is a common autoantibody found in almost all sera. Detection may require testing at 4 degrees Celsius or against enzyme-treated red blood cells to see reactivity. The production of auto-anti-I can be stimulated by microorganisms with I-like antigens on their surface, such as Mycoplasma pneumoniae infections. Anti-I is not associated with hemolytic disease of the fetus and newborn. The MNS blood group, this contains 46 antigens, nearly as many as the RH system. The M and N antigens are located on glycophorin A, and these antigens are destroyed by enzyme treatments such as Ficin, papain, bromelain, and trypsin. Capital S and little s antigens, so these are located on glycophorin B, which is a smaller glycoprotein similar to glycophorin A. Enzyme treatment effects, so they can be degraded by Ficin, papain, and bromelain. The extent of degradation, however, depends on the strength of enzyme solution, the length of the treatment, and the enzyme to cell ratio. Trypsin does not destroy capital S and lowercase antigens. Anti-M is often naturally occurring saline agglutinins reacting below 37 degrees Celsius. They are usually in the immunoglobulin class IgM and IgG. It does not bind complement. And for enzyme treatment, it does not react. For dosage effect, it reacts better with if you have a M plus and negative, so a homozygous capital M genotype. It's going to have that dosage effect where it's going to be stronger reactions for that. These ones are generally not clinically significant, so we don't usually worry too much about these. Anti-N is cold reactive, IgM or IgG saline agglutinin. It does not bind complement and it does not react with enzyme-treated red blood cells. Dosage effect, once again, it reacts better if it's an M negative and an N positive, and this one also is not clinically significant usually. For anti-S and anti-little s, so most examples of anti-capital S and anti-little s are IgG, and these are reactive at 37 degrees Celsius in the antiglobulin test. These may bind complement, and it results of the enzyme treatment, they may or may not react. It's kind of up in the air. Dosage does occur, but it's not as dramatic as the anti-M or anti-N, and once again, these are usually not clinically significant. The Kell blood group system. So this consists of 32 high prevalence and low prevalence antigens. For cellular distribution of the antigen, this is found only on red blood cells. It is not found on platelets, lymphocytes, granulocytes, or monocytes. So we have the K, which is the capital K antigen, which is called the KEL antigen, is one of the most immunogenic blood group antigens after the ABO and RHD antigens. This means it has a strong ability to provoke an immune response if foreign to the recipient. Because of its high immunogenicity, the presence of the K antigen can be a significant factor in blood transfusion and pregnancy. When a person who lacks the K antigen is exposed to it through either transfusion with K positive blood or during pregnancy, if the fetus inherits the K antigen from the father, this can lead to the production of anti-K antibodies. These anti-K antibodies can cause serious transfusion reactions or hemolytic disease of the fetus and newborn. Hence, we'll need careful blood matching for the K antigen and where it's important in transfusion medicine, especially for women of childbearing age. In contrast, the lowercase k antigen, also known as Solano, is very common, and antibodies against it are very rare because most people possess this antigen. Anti-little k antibodies are seldom encountered in clinical practice due to the rarity of K negative or little k negative individuals. Then we have the KEL blood group system continued. So we have the JSA and JSB. So the incidence of these, we have the JSB. It is a high prevalent antigen. It presents in most individuals, whereas the JSA is a low prevalence antigen. It is found in fewer people. For anti-capital A, so outside the ABO and Rh blood group antibodies, once again, anti-K capital is the most commonly encountered antibody in blood banks. It is important to note that anti-KEL is distinct, or capital K is distinct from anti-JSA or anti-JSB, but they all do belong to the KEL system still. For clinical significance, antibodies against KEL antigens, including anti-uppercase K, can cause severe hemolytic transfusion reactions and hemolytic disease of the fetus and newborn. These reactions occur because the antibodies target red blood cells, leading to their destruction. For immunoglobulin class, these antibodies are typically IgG, and they will usually react optimally at the antiglobulin phase, or the Coombs test, which is used to detect IgG antibodies bound to the red blood cells. KEL antigens are located on the glycoprotein that spans the red blood cell membrane. This glycoprotein structure allows the KEL antigens to be expressed on the surface of the red blood cells. The KEL glycoprotein is involved in maintaining the stability and function of the red blood cell membrane. For genetics of the KEL antigens, so the KEL blood group antigens are encoded by the KEL gene, which is located on chromosome 7. This gene encodes the KEL glycoprotein, which carries the various KEL antigens. Different alleles of the KEL gene can result in different antigenic variants, such as K1, K2, KPA, KPB, JSA, and JSB. The expression of KEL antigens follows Mendelian inheritance patterns. Then we move on to the Duffy system. So we have Duffy A and Duffy B. The most important Duffy antigens, like I just mentioned, are Duffy A and Duffy B. These antigens are glycoproteins found on the surface of a red blood cell. The presence or absence of these antigens defines the Duffy blood group system. So the distribution of Duffy A and Duffy B antigens varies widely among different populations. Some individuals may lack both antigens, where they would be Duffy A negative, B negative, which is common in certain ethnic groups, especially in populations of African descent. It does seem to be an association with malaria. So the Duffy antigens seem to serve as receptors for the Plasmodium vivax malaria parasite to enter red blood cells. Individuals who lack the Duffy antigen, so if they're Duffy A negative, B negative, they are resistant to infection by the Plasmodium vivax or malaria. This absence provides a selective advantage in malaria endemic regions, explaining the high frequency of Duffy negative individuals in such areas. So the Duffy antigens, Duffy A and B, are destroyed by certain enzymes such as physin, papain, and bromelain. Anti-Duffy A and anti-Duffy B antibodies. So these antibodies typically react best in the antiglobulin phase, also called the indirect Coombs test phase. Anti-Duffy A is more commonly encountered than anti-Duffy B. These antibodies are usually of the IgG class, which means they can cross the placenta and cause clinical complications such as hemolytic disease of the fetus and newborn. Some antibodies may demonstrate dosage effect, meaning they react more strongly with red cells that are homozygous for the antigen, so having 2 copies of the gene compared to a heterozygous cell. Duffy antigens are located on a glycoprotein that is embedded in the red blood cell membrane. The specific antigenic difference lies at amino acid position 42, which helps distinguish between Duffy A from Duffy B. The Duffy blood group system is controlled by co-dominant alleles, meaning that both alleles, Duffy A and B, are expressed if they are present. This results in the possible phenotypes of Duffy A positive, Duffy B negative, or Duffy A negative, Duffy B positive, or Duffy A and Duffy B positive, which means they would have heterozygous expression of both of those antigens, or Duffy A and B negative, so a null phenotype, which is often resistant to certain types of malaria. Then we move on to the KID blood group system. The KID blood group system is represented by the symbol JK and is designated 009 by the International Society of Blood Transfusion. The system includes 2 major antigens, JKA and JKB. These antigens are clinically significant because antibodies to KID antigens, anti-JKA and anti-JKB, can be challenging to detect during routine blood bank testing. So despite being hard to detect, these antibodies are common and can be a cause of delayed hemolytic transfusion reactions. The KID antigens are not denatured by papain or fisin. In fact, enzyme treatment enhances the reaction with anti-JKA and anti-JKB antibodies. KID antibodies have a notorious reputation in blood banking because they demonstrate dosage effect. It's a stronger reaction, once again, if they are homozygous for the antigen. They are often weak in reaction strength, and they frequently occur in combination with other antibodies, which makes detection challenging. The KID blood group system is genetically determined. The main antigens are JKA, JKB, and JK3. JK3 is a high-frequency antigen found on all red blood cells, whether they are JKA or JKB. Individuals who are JKA and JKB negative, or the null phenotype, also lack a JK3 antigen. So the anti-JK3 antibody, it is an IgG class. And for reactivity, it reacts with all red blood cells tested in a standard antibody panel. This broad reactivity is due to the high prevalence of JK3 in the general population. For clinical significance, it is associated with hemolytic transfusion reactions, and it can also cause hemolytic disease of the fetus in newborn. For implications, so individuals with anti-JK3 must receive blood from a JKA and JKB null or negative donors, which are extremely rare and hard to find. Autoantibodies in the KID system, so a rare occurrence of autoantibodies against KID antigens. These autoantibodies may be implicated in autoimmune hemolytic anemia. The Lutheran system is composed of 19 antigens numbered LU1 through LU21. Some numbers are skipped, so just keep that in mind. But some Lutheran antigens also have alternative common names, genetically determined by the LU gene on chromosome 19. For incidence, so Lutheran A is a low-frequency antigen, and it's found in about 8% of individuals, whereas Lutheran B is a high-frequency antigen, and it is found in 92% of individuals. Other Lutheran antigens vary in frequency, but most are considered high incidence, whereas the Lutheran null phenotype, or their Lutheran A and B negative, is rare. For clinical significance, it can cause hemolytic disease of the fetus and newborn. Lutheran antibodies are rarely implicated, but when involved, it typically can cause a mild form of hemolytic disease of the fetus and newborn. For transfusion reactions, Lutheran antibodies are typically clinically insignificant, but they can occasionally cause mild delayed hemolytic transfusion reactions. Resistance and sensitivity to enzymes, so they are resistant to fisin and papain, and they are destroyed by trypsin. For antibodies, anti-Lewis A, that is an IgM, and it reacts better at room temp than at body temp, and it will often go undetected in routine testing because most reagent red blood cells are actually Lutheran A negative. Anti-Lutheran B, so this is an immunoglobulin type IgG, and it is typically produced in response to pregnancy or transfusion. For genetics, the Lutheran blood group system is co-dominant. So we also have some antibodies, so anti-Lu3. This is a rare antibody, and it reacts with all red blood cells except Lutheran A negative and B negative, which are the cells that don't have Lutheran A or B. It is produced only by individuals who are recessive Lutheran A negative AB. Only a few specificities are commonly encountered for antibodies. For clinically insignificant antibodies, we have the M, the P1, and the I. These react at room temperature and are generally going to be considered clinically insignificant. But our clinically significant antibodies are our KEL, which is the big K, big S, little s, Duffy A, Duffy B, JKA, and JKB, so KID, A and B. These react in antiglobulin phase and are considered clinically significant.